So the ZUMA-12 is a phase 2 multicenter single-arm study where we evaluated axi-cel as part of first-line therapy in patients with high-risk large B-cell lymphoma. To be eligible for this study, patients had to have either double or triple hit lymphoma or large B-cell lymphoma with an IPS score of 3 or higher. In addition, they had to have a positive interim PET scan after 2 cycles of standard chemoimmunotherapy, typically either R-CHOP or R-EPOCH, and this was used as a measure of dynamic risk assessment in these patients. And the patients received Cy/Flu conditioning after the CAR-T product has been generated and received a single infusion of axi-cel. And the primary endpoint was complete response rate. We enrolled and treated a total of 40 patients on this trial, and the best overall response rate was 92%, and the complete response rate was 78%. And after a median follow-up of 16 months, 73% of the patients remain in remission. So as compared to prior studies, we observed similar adverse events such as cytokine release syndrome and neurological events related to axi-cel therapy, and there were no new safety signals in this first-line study. Now we also compared the phenotype of the CAR-T products in the ZUMA-12 study in the first-line setting with that of the ZUMA-1 study where axi-cel was given in the third-line setting and beyond. And we observed that in the first-line setting, there's a higher frequency of CCR7 positive, CD45RA positive T cells, and this associated with a greater CAR T cell expansion in the ZUMA-12 study compared to the ZUMA-1, even after adjustment for tumor burden. So overall, I think these results indicate that axi-cel can be highly effective in the first-line setting, and the T cell fitness seems to be improved in earlier lines of therapy compared to 3 or more lines of therapy, and further studies are warranted to investigate axi-cel in the first-line setting and to compare it head-to-head with chemoimmunotherapy.